Hey everyone, and welcome to Homework 06 answer key walkthrough. I know that Homework 06 was very difficult and challenging. That was the intent, and the idea was to give you a lot of different potentially useful things, like reading in multiple data at the same time, performing statistical tests, and also graphing. And there was actually an error with the type of data and also the code that was provided if it wasn't formatted, completely correctly. And so we'll get into that, but because of that error and because some people were having issues with that regarding the code that was provided in the original Homework 06, this part here where I show how to map gene IDs to gene symbols, I updated the homework online to make it easier for anyone who wants to redo it. If that part was giving you trouble and you want to have another crack at it, feel free to. The Homework 06 updated now is such that you don't need to have that because the data that's in there is a little bit different format. What the data shows here is that there's three folders like there was before, and there's an old. The old is the original data, and I have it here that the H3K27M, it's the same data essentially, and I'll show you what it looks like, but it's in a CSV file. And I'll go over what issues that were caused and everything at the end, but first I want to just kind of say, like, how do you, just go into the answering the homework and how to go about it, and also try to get a sense of, how I'd try to go about thinking about the homework and getting in the right format. And I think this could be useful, this part in particular could be useful for any coding assignments that you have in the future. So if we go to the updated homework, I have it listed out a little bit differently, and because of that, you don't need to map. So the steps are to read in the data for the genes of interest, and any gene that you want to include. If you didn't want to include, that's fine. Perform a t.test to see if there's a difference between the TPM expression values between the wild type and H3K27M samples, and then create a graphing function to create a box plot for each of those. And so a good way to start thinking about this is just, what is the data what does the data look like now, and what is it going to have to look like when we get it in? How would you want it to look? So if we go into the files, and so here's an example of the original data, the .csf, and because it's a tab delimited file, you could open this in Excel. It opened it up perfectly fine, and this is what it looked like. For the rest of this, I'm going to just use the updated data. So basically what I did is I read in all the data, and then I wrote out a mapped version of it where it has one gene symbol already, so you don't have to map it yourselves, and then the corresponding TPM value. So, and they're a little bit smaller data sets, which might be helpful for those of you who didn't necessarily have the best computer, because depending on how you did this, there could definitely be situations where it would be difficult to read in all the data at once, or kind of a slow process. So let's just take a look at the CSV files that are the updated versions that I provided now. It's the same idea, just it doesn't have all the columns. It does have a gene symbol column, a transcript ID column, and the TPM values. It doesn't have the number of reads or the gene length anymore, but it does still have sort of the same sort of problems that we're having here, where ideally, let's open that one and one more just so we can look at it, where the gene symbols, if we put it side by side, the gene symbols and the transcript IDs are the exact same values for in all the files, in both the wild type and the mutated setting. But the thing that is different, obviously, is this TPM value. So one way you could think about doing this, and this is the way I asked for in the original prompt, but you could do things differently. So we'll get to the way that one way you could do this would just be, I just want to take basically this column from this data set, the next one, and be able to put it right next to this one. And obviously though, then I have two columns with our TPM, and it's like, well, obviously I would want to rewrite that as well. So maybe if I wrote this as BT245 TPM, like the TPM values for that sample, and then this one as this came from the dip GVIC30. And this is kind of where I was going in terms of having, at one point in the original homework, I mention if you have basically the first column be the gene IDs that contains the transcript IDs, and the remaining columns would contain the TPM data. This is kind of what I was getting at. However, that's one way you could think about putting the data together. You could also think about putting it together by the rows and just saying, well, here's the data for, BT245, and so maybe if I get rid of this and, this is the data for BT245, and like putting right on top of that the data for the DipG or DipG6C30. So either way would work, and actually the other thing to consider, not just like how it makes sense just viewing it to put things together, but the other way is to think about what you will need to do with the data, of course. And what I asked you to do is a t-test and also graphing. Well, with the t-tests, how to format the data and everything, one thing that would be helpful to do is to run like just the question mark t-test and look at the help page. And there's basically two ways of running a t-test. One is it says t-test x, y, and you could, read through the arguments and see what they're supposed to be. But basically, x would be a vector of values that corresponds to one group. So, if we were doing the height of male versus female people, right? So if we're doing x is the height of, female people, we can say like, oh, here's just this a vector of heights for each of the female subjects in the data set, and here y would be the vector of heights for the males in the data set. In this other version of it would be to use a formula. So formulas are something that didn't really get introduced, but that would be something like that would look like this: height by, I read the tilde by as a by, and do height by gender would be a formula, for example. So basically, if you had a vector or a vector of values that were all the height values. Here are all the height values. So, it could be sorted by gender. It could not be sorted by gender. It could go male, female, male, female, male, female, whatever it is. So long as the gender vector that you would give it is appropriately sorted. So that'd be, if the heights were like 6 feet and then 6 feet, but the first one was male and the first one's female, then the gender vector would go male, female. So instead of separating them into groups, you could just have it say, oh, this this gender vector points to what type of value this is, or what value the height comes from. You can actually read the formula here. They show it as LHS by RHS, where LHS is a numeric variable giving the data values. So in this case, it would be just all the list of height values. And actually, in our case, let's just talk about it with TPM values. It would just be the expression value for those. And then RHS value is just a factor with two levels giving corresponding groups. So in our case, it'd be TPM by H3 status, for example, might be a good formula to write. And so it would be, here are the TPM values but you'd want to do this, for each gene, right? You'd have to filter for each gene, then do TPM by H3 status. So, and there's also a tidy way to do this, where you don't, I already said kind of for, and that's the way that potentially in base R, and it makes the most sense to use, but there's actually a tidyverse way of doing it that is very efficient in terms of lines of code, but I'll show that off to you later. Of course, that is the way of doing it. So in that case, right, what we'd want really is we'd want, if we were looking at genes, we wanted to think about this in a tidy format, right? Tidy format is, again, every variable gets its own column. And if you were to put these two together, like we had it before, well, both of them are TPM values, right? Both of these columns, even though there's TPM values for this model and there's TPM values for the other model. So this is not in the tidyverse sort of language. If you're going to use ggplot, like this isn't the tidiest way of having the data. It could be an efficient way of having the data, having them all in columns, and each column being a different model. But other way to do it would be having to think about it as just like all right, well, I will get rid of this one, and then say, okay, well, here's this list of TPM values, and now I know that, what is the other variable here? The other variable is the model system that we're using, and so that would be to say something like BT245 is the model, and so and of course, in Excel, it autofills it this way, but that's fine. We're not actually using this in Excel, right? And would be to say, oh, I have this model column of what the model is, and maybe also that H3 status column, so we know that what the model is here, and this would be, since this came from the H3K27M folder, it's H3K27M, and you can autofill that. And then it would be to have, model DIPG VI underscore C30 underneath it with all of its values. So again, be more stacking the rows on top of each other. So I will show how to do it kind of both ways, a column version and a row version, but once we get to the graphing, we really are going to need to have the data in a tidy format because, of course, using ggplot. You could do it in non-ggplot, and then and that would actually and again, it would be essentially the same thing of what you would do, but since we pretty much only graphed in ggplot, that's what we're going to do. So first step is to read in the data. So I have two different ways of reading in the data here. The first thing is really useful thing is to use this list files things. And of course I call the library tidyverse because I'm used to using tidyverse functions. And what this list file does is I point it to a folder, and pulled it into the H3K27 folder, and I ask for the full names. And what this does is it gives you the dot, RNA-seq, blah, 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 and it gives you the whole name of that file. What that's going to be important for is to use, if I'm going to use it in a read function, I want the whole name of the file, right? I want to know that it's here, so I can just say, read this file. And if you know how you would write a read file, CSV. So the way to kind of do this backwards would be to consider what read CSV would need. Oh, if I need read CSV, it would be file is equal to, and then you put the quotes, and you say, it's dot is just this directory, and then do RNA-seq process, and if I wanted to get the old stuff, I could do that, and let's say I wanted to read that file. It's not a CSV file, so this wouldn't work, but right, that's how it would look to begin with. So now that I got this list of using list files or a vector of all the files that I want from H3K27, and I have a wild type files as well. One thing that might be nice, especially if I'm going to use this, put them together, right, is to just know. So I got rid of the Excel files, but right when I was talking about putting them together, one thing I said is that you'd want to know which column is coming from which data set, and you could use this, but it has all the like the .RNA-seq and everything. So instead of doing that, is what I do is I say list files again. So I'm going to get the list of if I run this, I get this list, but full names are not equal to true. So it's just the files that are in that folder and their names. And this is the name of all the models, and so if I wanted to keep track of them, one thing that might be nice is having a list of it. And so, but they all have the .csv at the end, and that's probably fine. It doesn't actually matter whatsoever. Like the unique information that identifies them are right there. The .csv is not important, but I can substitute that out if I wanted to be pedantic and say, so I take that vector that was created here, I say the pattern is equal to csv, .csv, replacement is equal to nothing. And now when I do this, and if I look at the H3M models, I get rid of those that other stuff, and I only have the model systems. And the model systems, of course, are nice in that it's listed in the same order. So it is, that is to say, like the first model system here would correspond to the first file in H3M files. And of course you could do that for the same thing for the wild type models. And then I mentioned using sapply, but if you use sapply, there's a function actually that you'd want to do simplify is equal to false. And so really you just want lapply. So my apologies, I was actually used to using sapply and simplify is equal to false. So that would work, or using lapply. It's just the sort of basic, essentially sapply with simplify is equal to false is the same as lapply. So, but what this will do is it'll read in all the data, and it will assign the first list item to be the first file. So if we do look at the list.dipg and we pull out the first value of that list, which is, you use double brackets for that, you can see what it prints out. It prints out a tibble because I used the read underscore, so I'm using the readr functions, so that converts it to a tibble, and then it's going to have this table where it's gene symbol, transcript ID, and TPM, just like how we saw in the Excel files that I was showing before. So that's good. The one thing to also keep in mind is that so we're using lapply, and we're applying basically the names of the wild type files and the H3 files. I put them all together by just using C into read CSV. One thing to keep in mind is, of course, we want to kind of keep track of which files are wild type and which ones are H3M. And so it's important to note that I did a wild type first and then H3M, the H3 mutated files. So that gives me a list of DPIG1, and it's a list of 45. If we take a look at the list values here, it's a list of 45 different elements. So all the files, all 45 files. So seems to work well. Or you could do this. So I show using list files is super useful, so I used it again, which H3M files, so this is the same thing as we saw before. But one thing you might want to do is say like, oh, well, I already have the files names, so I don't really need to use list files again. I just really want part of it to know what the model is. I just want to know what, like, if I were to be able to split the file up, I just want to kind of know, the one section of it. And so what I use for this is a string split, which is another thing it's actually the stringr function package, which is in the tidyverse. There's different ways of doing this. If you did it in base R, you'd have to use an apply method. But one thing I do is do string split. The string is H3M files, so the vector that was here. The pattern is going to be /, which means the path. So it would kind of, any folder that's there is going to separate by that. And I'm also going to separate by the period. And the reason I'm doing a period is because, again, that last .csv is, I don't really want that .csv, so I want to separate that out as well. And so what you'll see is let's just print off the H3M files to see what they look like again. So you can see Anytime there's a dot or a dash, it will separate them into different groups. And kind of just want to show off this functionality. So if I do string split and kind of just show you what that does. Oops, I should not do the fifth column. So what simplify does is usually it returns a list, but lists are kind of annoying to work with. So what I did instead was return a table with simplify is equal to true, and that's a nice thing that you can do with the str split underscore split. And you can see that the first thing and the second thing are there's nothing there because it didn't include the period or the first slash. You have the RNA-seq processed, H3K27M, and what the model was. And so really I wanted that fifth column, and that's why I do the comma 5, and I'm able to get the whole thing here. So if I went through that, then to get the list of files and the models just like before, but in this case what I did instead was I wanted to use a for loop just to show you that it would be equally easy to do a for loop. Before a for loop, right, you want to put values into a for loop. You always want to, before a for loop, you want to create a vector, an empty list to kind of store the data in, to say like the ith element of this list is going to be this, or the ith value of this vector is going to be this. So first is to just say list dipg2 is going to be a list. You saw that dipg came up in the name of some of these values, so to explain that, a lot of these there's a mix actually of glioblastoma and also DIPG, which is actually a common or more common in children cancers. It's diffuse intrinsic pontine glioma. So I renamed some of these things, and we'll get to that in actually later. But really, it just refers to the values that we're looking at here. So that I just wanted to make an empty list. And then use a for loop, and you can see that I'm doing it for I in 1, 2, which is a very common thing. Usually you want to have it programmatic. You want to say what it ends, so if you did add any wild type or H3M files to this, it would still run well. And so for that, I'm going to do the length. I want to do the length of a vector, wild type files, and H3M files. So put those two vectors together, concatenate them, and do the length of the whole thing there. So really this would be the same as once it evaluates, it's going to be for I in 1 to 45. But, again, if I put in more wild type files or different H3M files, it would still work correctly. And really then I just want to say the ith value of the list is a read CSV file is equal to C wild type files, H3M files, I. So this is going to also put in a list of files in the same order as before. And we can actually make sure, like I have a list DIPG2 and list DIPG1, and there's a function identical. X and Y would be list.dipg1, list.dipg2. True. Great. So we were able to read in the data in two different ways, a for loop and apply. So now it's time to combine the data into one data frame, and I'm going to show the two different approaches. One is to combine by the rows, of course, and one of them is to combine the columns, as I was talking about before. And columns was the way that I suggested in the last homework assignment, but because it's mapped already, you don't need to do that. I'll show both ways equally well. So in just binding the columns, it will work. I'm using the bind calls from tidyverse. It doesn't really matter if you did the cbind function would work, which is the base R way of doing it, would also work. And then I'm going to select one and anything that contains TPM. So really, right, like I said, I know based on how the data is formatted and everything, every single column, every single, if I were to look at like list DIPG1 and say 1, and I want that first column of gene symbol, let's actually do the identical thing again. Are these two vectors identical? List DIPG1 gene symbol and list.DIPG1 with the second value. So basically the next file. And it's true, so that means that basically the gene symbol, and what I was getting at is the gene symbols for all of the files are in the same order, they're the same values, and I showed you here, at least with 1 and 2, there's probably a more robust way of checking, but that they're all identical. So that means if I just bind the columns together, really anytime there was a gene symbol column, that is really just repeated information. It's the same information every single time. So what I want to do is bind the columns, but then I really just want to take the first gene symbol. I could take a transcript ID as well if I wanted to maintain that information going forward, but I don't really need it. And then, because all of them were named TPM actually, what I can do is just say, and also how in the tidyverse, you do get renames of anything that where names repeat, and so TPM ... 3 refers to the fact that it's the third column that it's putting together, that it's binding in the columns, and then gene symbol 4 refers to that it's the fourth column that it's binding. So even though it's the second, this gene symbol to gene symbol ... 4 is actually the gene symbol for the second DIPG1, list DIPG2 here, the second data frame in the list. So basically, here's the first data frame, which would be these 3 columns. The second data frame would be these 3, and it just numbers it by the column number. But that's convenient enough because it is just putting a ... 3, so I could just be like, okay, if it contains TPM, that's the value that I'm interested in, that's the normalized read count values. So that is the value that I'm going to be able to compare more easily. Now, if I do that though, I get this, let's do a view of this. I get gene symbol ... 1 and TPM ... 3, 6, 9, 12. It's going up by 3, so it's getting every single one. There's 3 in every value. So that's exactly what I wanted, but of course I don't know what 3 referred to, or 6 or 9 or 12. I don't know what values those referred to. But this is where it comes in very useful to having the wild-type models and H3M models, and also having them in the same order already. We already know that the first wild-type model is the first wild-type model that I have listed here. Wild-type model, 1840. Well, if again the wild-type files and how they were listed in 1840 was the first value. So that is the first value of the list as well. So I can actually just rename the columns and do column names and of DIPG1 and assign that to be gene symbol, the first column, and that's fine. That's basically what we're looking for. Wild type files, because then it's 1840, then 2230, then blah 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 blah, and then H3M models eventually, like once it runs out. So now I can do that, and if I take a look at this, now I can see which models are taking up which columns, and I know that they're all the TPM values. So that is one way to make the data frame, but you could also bind the rows, and there are the easiest way, you just have to bind the rows, and you can see that you get a value here of 688,000 1,000 rows on top of each other. And this is actually one of the reasons why some people who bound the rows with rbind in the previous one, where there was more values, if you did it in the original homework assignment, you probably got a data set of about 10 million different rows, and so that could make your computer kind of stall potentially because it's a lot of rows. If you're going to do it in base R, so bind underscore rows works for whatever reason. That's the tidyverse function. If you use rbind, you'd actually have to say do call what is equal to rbind arguments is the list of that you're trying to bind the rows of. In this case, I think it's because of how rbind works and the fact that you're binding a list, and then you want to bind the first value of the list, the rows of the first value of the list with the second values of the list, and then, you know, bind the third onto that, and the fourth onto that, and the fifth onto that, right? So it can be a problem if you do rbind, because you need to kind of, by itself, because it needs to kind of, in a way, put them all together, not just like, you know, two of the values together. So that is to say, you need to put 45 different tables together, whereas rbind might just put two of them together. That's my understanding of do call, or what it's basically the function of do call here is. But again, bind rows actually works really well, and if you're going to look at it, you know, it just looks, oh, hi, this is the rows example. But of course, you know, I don't know if I were to scroll randomly to the middle. I don't know what is this value from, or what is it is this ACTA1 from H3 K27M or a wild type model? And so we would want to sort of mutate the rows. And how I did this is, again, you can use the fact that we know that the first column, or that basically the first value is going to be that wild type model. It's going to be that the first several columns are going to be 1840, and then when the columns for 1840 end, it's going to be the columns for 2230, and then when the columns for that end, it's going to be the BTB blah 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 blah, etc., etc. So what I can do is I can just repeat the value. I'm going to say repeat the wild type model name, basically, that we've seen before. And then, of course, repeat the H3M models, so I put them together. At this point, maybe it would have been better to just put the models together and be like into one vector, but I like having to see the difference between which ones are wild type and which ones are H3M. And in this case, I want to repeat it, and you can do it in different ways. The base, if you were to look at rep, the sort of, nope, just rep, please. Sometimes it works for you and sometimes it doesn't. So if you were to say rep, it shows a vector, and you can either do the number of times, basically, that it should repeat. And so if you were to repeat like the vector 1 through 4 and twice, you know, it'll show you 1 2 3 4 1 2 3 4. But if you do each is equal to, it will repeat the values 1 1 2 2. And so this is what I want to do. I want to repeat that first, you know, 1840 value, and I want to repeat it, well, for the number of rows that exist. And fortunately, the number of rows for all of these values, or all these vectors, are the same, or all these tables, the original tables, are the same. And so for that, I can say, well, how many rows are there in the original list DIPG2, or so basically for the file that was read in and put into a list, how many rows are there? There's apparently 10 plus 15,292. So take the number of rows there and repeat the wild type model, or 1840, for that many rows, and then go to the next one and repeat that one for about 15,300 rows, and then, you know, keep going until you run out of rows to do, and that should fit exactly right. And we can kind of check that if we wanted to repeat this, we can say, well, what is the length of this repeating thing? Apparently too many parentheses. It's 688590, and when we bound the rows together into this rows example file, you can see the rows example has 688590. So we are getting the same, repeated exactly for the same length. So that's good. And it's going to fit it in correctly. And then you could do this different ways. You could probably do it with repeat again, but what I did is say mutate, create a new column then, and then say h3 status is equal to, and do an if-else statement. So in this case, I'm using a kind of if statement that a dplyr if statement, and this if statement goes if the model name is in wild type models. So if we have a list after this first piping operation, we'll have a list of the model name in 1840 going down, and if that name 1840 is in the list of wild type models, repeat and just say, wild type, or if it's not, then do K27M. And in this case that works because it's either wild type or K27M, right? There's no third option. If there was another option, then you would have to potentially do another if-else statement in here, which happens quite often, and you can get like cascading if-else statements by saying, well, if this is true, do this, and then check for something else if it's false, like if-else, and do another function. And then, but in this case we don't need it, we just need to assign wild type or K27M. And so once we go through this, DIPG2 would look like this, where it has that wild type setting and everything, and we can well, sorting will take a long time because of the fact that there's 6,850 rows, but then if we go somewhere random, let's say to this one, we can see that it's a wild type one, and if we were to print out the wild type model's name, that is, the last one here. So if I were to just scroll down a decent amount farther, probably, there are, so many rows between them, but hopefully if I scroll down long enough, I will find, oh, it did sort actually, so that's why. But let's just scroll down randomly. So now I sorted by K27 status, and you can see that there's the PS11329, and if I were to print out the H3, please, H3K or H3M models, you can see that the PS113293 is down here. So it looked like it assigned it correctly. It's always nice to check these type of things, make sure there's not a problem. And now we need to filter for the genes of interest. And this is actually done, I do it the exact same way for either model of either thing, and I'm going to just be explicit. This is the column example, and this is the row example. So now if I do that, you can see that we have a lot fewer for the columns. We have a pretty efficient column column and row count. We have 22 observations by 46 columns. Rows, of course, are going to be a lot longer because the gene repeats every time, but we have 5 columns by 990. So, but essentially we have 2 tables that have the same thing, same information. So now doing a t-test. This case I'm going to use a for loop, and basically, right, in the first case we talked about a t-test, and you can either say, here are the values of x, here are the x values, and that is the vector of one group, and here's the y values, the vector of another group. Well, the nice thing that we can do here is we can create this like genes H3M or genes wild type, and what this does is it takes, I want the column DIPG data that we have, and I want the columns specifically of the H3M models for the genes H3M. So I can sort them this way, and because the names are, because the name of H3M models are these in parentheses and everything, it works really well in sort of a base R setting to just say, I want that column, and you can specify a column when you have a data frame as either the name, so that here we can look at it this way. You can say, if I wanted the first column, right, I can say do it that way, or I can do it this way where you put in parentheses and you say gene symbol. You get the same values out. So we can call those columns using H3M models, and we can call those columns using wild type models, and then all of a sudden we have a and I did it as matrix because sometimes, I don't actually know, I didn't test this, but sometimes because tidyverse really likes to have when you take values out of it, the values to be in a tidy format. So whenever it's a tibble, basically that it always remains a tibble, even whenever you use a subset. And because t.test, you want a vector of values, which is the more default. If you have a data frame and you just use one row, it will essentially be a vector instead of, again, with, right, when I just did one column here, it said it was still a tibble. So I didn't necessarily want to deal with that, so I just made it a matrix just to make it make sure that issue wouldn't happen, which is something you just kind of like learn to kind of think about. So if you look at the genes H3M, I have every row is still a gene. I just don't have it labeled here because I don't want to have that column be included in the vector values, right? I don't want to have a non-numeric value there, and I like the fact that you can see that it's a numeric matrix, that it only has the numeric values there. And so basically I'm just going to do a t-test between the values that I have from this one matrix that has the H3M models and this other matrix that has the wild type values, and I'm going to do that, and I still use broom in this case because it's the value. If you just do a t-test, so if we take, oh, and of course you need to have a list, empty list to put the values in and say the ith value is going to be that. And so the reason why is if you do a t-test, and let's just store it as an example, and you do a t-test, and I'm just going to actually copy this code here. So this is going to be the last t-test that it did. Right, so in this case, I is set to be 22, so it's the 22nd gene in the list. It's going to, or subset to the 22nd row. If you see that it's an example, and so it's a list, and so if we print out example, it prints this, which is nice in a way, that you can see this whole example, tells you the p-value, degrees of freedom, and everything, but it's a little not nicely formatted. And so the broom function, the idea of it is to sweep things into a tidy format. And so we do broom of the example. You see that basically all of these values, statistic, parameter, p-value, and etc., just become their own column. You have the estimate, estimate 1, 2, statistic, p-value, etc. So I do use tidy to kind of cheat to make a list where it's already a tibble. Now I can r-bind them, or I could have just used I didn't have to use r-bind, of course. I could have just used bind rows, but bind underscore rows, the our dplyr functionality for this. But I just wanted to make sure that I showed you the do call and r-bind variant of this. So then I have a t-test table where basically I took the list of results, t-test results full was a list, and bound those rows together into a t.test table that I have here, which is nice. It shows you all the p-values and everything, but it doesn't actually show you what row 1 is and row 2 is and row 3. And it was 1 for every single gene, and the genes were in the order that the values here. So I made this table with symbols, which is not very, maybe I should say t.test table with symbols or something along those lines. But take t.test, I mutate so that the gene symbols are going to be from the DIPG data, the gene symbol values, because that's where it's from. All these are from, and I want to select. What this does is that, if you notice, it says everything, so it's selecting everything. So what's the point of keeping everything? Well, in this case, I actually am just reordering the columns, and this is how you do it in the tidyverse, is that you say select, and you select what one you want to have first. You can say the second thing, the third thing, and then say everything. Basically, we'll do gene symbols, and then put everything after that in the same order. Table with symbols, and then I call it do knitter, so that if I was going to publish this as a GitHub document, that you could see the table nice and neat here, so you see the values and everything. Cool. So that was how to do it in a for loop. Pretty standard for loop in this case. A couple of tricks there. This is actually easier in a way, less code to do with the row t-test. So group by. So group by, what it does, it basically makes, you can think of it as putting a lot of smaller data frames on top of each other. So it's super useful because what you can do in this case is say, how the rows are set up. Let's look at the row DIPG data here. How the rows are set up, right? They're gene symbols, and the gene symbols repeat. And so, I kind of want to group it so that all of the gene symbols are grouped together, because I'm asking for every single gene symbol. So group by the gene symbol, and then what that does essentially is take and it breaks it up into 22 different data frames, because I have 22 different gene symbols that are just kind of stacked on top of each other. So you can kind of still use the same coding and everything, but you can do also some other summary functions, which I think we've covered in the course. But one thing you can do is basically do a t-test where, say, the t-test, the data is equal to dot, which is the grouped row DIPG data, and the question I'm asking is, of course, TPM, does that change based on H3 status? And of course, I don't know if I talked about this in terms of, ideally, if you want to actually know if H3 K27M is affecting gene expression and everything, a better controlled experiment, of course, would be to just mutate, like have the same cell line and mutate H3 to H3 K27M, have a wild type, and do expression analyses between the two, so that you can have it very nicely controlled. So this is more of a correlation, because of course there's other differences besides just H3 K27M between these two samples. So this is more investigatory, maybe. Maybe there's just like, oh, well, we have these samples and they're sequenced, and they're different. We have this H3 status difference. So let's see if any of these genes that we're interested in are different between the two. But anyway, so that let's go back to the code instead of the science. Right, we want to say, does TPM differ by H3M status? But because it's grouped, it's actually going to do a t-test for all of the different different genes. It's going to do that because of this do and tidy functionality. So tidy is going to do the same thing as before and make, well, in this case, it's kind of just going to force the t-test to work in a tidy, using with it being grouped and everything. So we run that, And here I just have it printed out an example of how this works, right? So I took the row DIPJ data, if I group by gene symbol as before, and then you can count the H3 status, for example. And here you can see, it just counted the number of, here's ADARB2, H3 status was K27M, there's 23 samples, and there's 22 wild-type samples. This is going to be the same for all of course. And it just basically counted those values, but don't need that code for, that was just for explanatory purposes. And now if I were to just print this table out, run the whole thing again, you can see in this case, because it was grouped by, and the default sort of grouping parameter again is doing alphabetical order, so it's actually listed in alphabetical order, not how. So the only difference between this table and this table is that trying to scroll is not working very well. So the only difference is that the table that we did with the for loop went in the order of how the genes were listed, which was the order of the vector that we gave it in the first place, I believe. Actually, no, it wasn't, but it was just based on the order that was filtered then. So the order that it existed in the data set, whereas this one went in alphabetical order. But otherwise, two ways of doing the t-tests. There's not really a good way of doing the graphing function, but it's having, again, in a longer expression format. With the mapping, that it might be more efficient to have put it into columns and then pivot it. So I think we've talked about pivot, and I don't want to make this too much longer, but the code is here. Basically, we are pivoting from the column data. So again, take a look at sort of the before and after here. Not what I wanted to do. The column data and long expression. And you can see that it's pretty much in the same format that we had the other values in the row when we bound the rows together at the end. But basically what this does is it's column data, and it's saying keep the gene symbols, but then for everything else, take the name that's here, that is the column names, and put that into a column called model, and take the values that are in this all over here, and make those values equal to the expression values. So it pivots it longer, and then it also puts that if-else statement, and in this case doing model, if it's an H3M model, it's H3K27 H3K27 mutated or wild type. So, then basically what this would have done, the pivot makes this, so these three columns here, gene symbol, model, expression of those values, and then also that last mutate puts in this last column saying, is the model wild type or not? So then if we wanted a graph, because we're going to do this multiple times, I'm going to use both a function and a for loop. I want to make a function where I plot the data. I'm going to make this value plot inside that to be. So let's go through this with a function. So a function, you define the arguments, and in this case, I want the function to have the argument dataset and the argument gene. So what it's going to do is it's going to take the dataset that you provide. It's going to filter for the gene symbol is equal to the gene that you provide, right? And so basically we want to take this long expression value, and we want to just filter so that you have one gene at a time. So just filter for BRINP3 and not all the genes. And then it's just going to plot those values. It's going to say X is going to be H3K27, which is this column here, the wild type, or whether it's mutated basically. And then the Y value is going to be the expression values in TPM. I also made a fill so that just color coding, so one group is going to be colored by H3K27M status. I wanted to make a box plot like I mentioned before. I'm changing the theme because I like to change the theme. this is one thing I did want to mention in terms of the labels. So one thing that you can do for graphing is, because it's a function, I kind of want to take this gene value that I have here and label each graph with that gene. So what I'm going to do is I'm going to paste that name. So the gene is, again, what's going to be provided, and say that gene, BRINP3, for example, and then I put BRNP3's expression in GBM models by H3K27. Maybe I should just say, here we'll say glioma models by H3K27, a mutated or wild type status. And then I wanted to center the title and get rid of the legend. One thing that is important, if you're going to make a graphing function, one thing you need to do is print, at the end, print the plot. You could wrap the whole thing in print if you wanted to, but you need to print the plot, otherwise it won't actually print when you do a for loop. That happens with a lot of graphing functions. Actually is just the need to be explicit and say, yes, please print it once you're done making it. So I'm going to create the function just by hitting Control-Enter, and then I can just make a for loop, and I know what genes I want. They're the genes of interest. So I'm going to do for I in 1 to length genes of interest. Gene grapher, I'm take the long expression data set, and so the data set is going to stay the same every single time. But what gene that I want? Well, I want to take the first value of the gene of interest, or the second value of the gene of interest. So now I can create that for loop, and you can see that here we have IRX1's expression in glioma models by H3K27 mutated or wild type status. There, BRD2s, and you can just kind of scroll through the different graphs, and let's say, if you wanted to take this analysis further, then potentially you could print on there you could use the data that we have in this table here and print the p-value or what have you up here. Technically, these should be adjusted for the fact that we did multiple tests. So, we did a test for every single gene to see if it was differently expressed. And so ideally, you'd want to do an FDR correction or something to correct for that. But, that's getting into if this was actually going to be used by someone to sort of look into things. And so you can see that sometimes the expression doesn't really change, but then sometimes, you see, slight expressions that might maybe attributed to H3K27M and maybe are worth looking into in a better controlled model, or something like that. So, and then of course the last thing to do is print the session information so that everyone can see what packages that were attached and what was used. All right, well, that concludes the answer key portion and kind of going through and everything. I will take the moment to just give a mea culpa about what happened with the expression data. So one thing you can probably tell is there's two things that happened. One was there was a mistake in the code that I provided here that that it didn't do exactly what I was thinking, but it's partially because I changed the data sets at the last minute, which is why you can see, for example, things like gene ID when there were transcript IDs, and GBM when it's there's both GBM and glioma in this data set. So if you had formatted it like how the homework 06 asked and did, that the columns thing that I mentioned of using transcript IDs in one column and the remaining columns containing the TPM data, you can see what it would work. It would give you values like this out. Actually, well, obviously the gene IDs are not mapped in this case. was printed off before. But what is going to do, and I don't have anything loaded, so I'm not going to run through this whole thing, but the problem is that the sequencing that was done in these set of experiments was much deeper than the experiments I originally used for the homework and had to get rid of because of that they're not supposed to be publicly available. So the genes that I used in these experiments actually were sequenced deeper, so there was more genes that overlapped. There was actually quite a few duplicated gene entries, and there was long non-coding sections and things of that nature where the gene symbols weren't actually very. There would be problems if you mapped it. The first problem is if you were to use this code, or the code that's provided here. That is the first problem. The second problem is that when you have if you were to use a rows thing, that wouldn't really work because of this function here, right? It removes the duplicated gene symbols, and this was fine for the Verdil dataset because it wasn't sequenced as deep and didn't have the different isoforms for every single gene. This dataset actually has quite a number of isoforms if you go to the original data for every single gene. Like EGFR has 3, like 8 or so actually, but like 3 protein coding ones and like 5 non-protein coding ones. And that was something I didn't realize when I did the data. So what happened? So let's go through the first thing. If you were to combine the rows and everything, you can see that this is removing duplicate gene symbols. They were removed instead of average because they usually don't correlate particularly well. And that was just to make it easier to have one value for things. But if you were to combine the rows, then of course you'd be getting rid of basically all the rows, right? If you did this mapping thing, you'd still expect, right, in the original data that I mentioned, there would be 10 million rows. If you were to, combine all the rows together, if you were to do this type of thing, you'd get back out like 1/20th of that values. 1/45th of that value, actually. You just keep one data set because it would remove all the duplicated values. So that would be one error that, why this ended up not being the best code for everyone, depending on how you put the data together. The second thing is actually that this actually, when I used this, like I said, from a different project, and because we subset the genes here, you don't really need to have whether or not they're duplicated even in the first place, right? Because ideally that these wouldn't be duplicated and they just have one value. But because they were sequenced much deeper, it's unclear that this would select for the gene that we're interested in. And then if you didn't use it, it would probably have it so that you'd have multiple genes. So IRX1 would have like several different expression values. And so then when you were doing the t-test, again, it would work, but it just kind of like lost its meaning in a way because of not knowing that it's the main transcript for things. And so that is why, so I do apologize for that, for not realizing that was an issue. I ran through the code and everything, and this worked, but the problem was because of, again, because it was a deeper sequencing, that there was actually multiple genes, and so they in a way, the gene, like saying this difference of expression had less meaning because there was like multiple different transcripts, and whether or not you should like average them together, like there was a lot of different questions that it kind of came up with it. So I potentially didn't explain that very well, but it's not super important. The main thing is that if you're curious what happened there, I edited it with this script, and basically what I did was I found this main values, and so if you were to look up main match annotation of NCBI and basically ensemble data sets. And what it does is it actually, the idea is that you have this main select, and there's it selects the sort of prototypical or the most highly annotated, and also the sort of when you have multiple transcripts and everything, it basically there's a select one, this main select value that is going to be the standard expression for this one. So for the gene, so the one that is going to be probably the most expressed for those values, or the one that you kind of associate with having the most functionality, because some transcripts don't even function all that much or don't do really anything. So that is what I used. I basically downloaded a data set and filtered for those select genes, so that now that when I do the data, and then I just provided it, like I said, because having you do your own mapping, I wanted to include it in case that mapping was going to be useful for anyone. It took a long time for me when I was first starting to look at the data sets to learn about these libraries, and to be able to kind of just figure out, oh, I just want to map the gene transcript information over. So that ended up being the solution, was just to provide that data and sort it so that there was only one transcript ID basically for any of the genes of interest, so that you can answer these type of questions reasonably well. Anyway, I hope that in general everyone found something useful about the answer key, that if you take the time to go through it, and there's a lot, it was a lot to ask for the end of this class and everything, for this as being homework 6 and everything. We did not expect you to be able to do this really, but we did want you to start seeing what you could do, given enough time and effort and everything, and what sort of some example of what kind of can make coding so useful. So thank you for making your way to the end of the video, and I will actually I hope you have a good rest of your academic year, and hope to maybe see some of you around. Take care.